Are there any questions before we get started? Last chance? Okay. We want to finish up a little tiny bit of eukaryotic gene expression regulation, then we're going to move into GMOs. By the way, who was able to come to the talk on Friday? Wow, fantastic. So almost the entire class. Great. I'm looking forward to grading all those extra credit submissions. Okay.
By Friday, correct?
Yes, due Friday, this coming Friday. This is the third time that I've brought this topic up after we heard, I think this is the third time, after we had a student presentation on this topic, circadian rhythms. So there's one thing that I forgot to mention last class I wanted to be sure to mention just because it's relevant to something we've learned already, splicing. How this point mutation, remember, the point was you can have a single mutation here where there's a sequence of a gene and there's two nucleotides on top of each other here, A and C. What that means is one's the original, one's the mutant version. And look at where that mutation is. Here's exon 11 in the shaded area. What does that mean this is? This is intron between exons 11 and 12. And that mutation is right next to what? The splice site. So what this mutation does, I know it's not in the splice site, it doesn't affect the G, the T, or the G and the U if this was translated into or transcribed into RNA, but it's right next to it, and it actually does affect splicing, and that's what was shown. The original presentation showed you this figure, but we didn't go through it in detail, that when you have that mutation there, that causes an alternative splicing event that causes either exon 11 stays in the protein, or if when this mutation is present, that exon gets removed. You don't have exon 11 in the Cry1 gene, this gene that's important in establishing our circadian rhythms. And that's what that single nucleotide mutation does, right? One nucleotide change, but it affects splicing of this gene, and then you start being a night owl instead of a morning person, right? One nucleotide change, alternative splicing, and a change in behavior. Now, of course, this is not the only mutation or the only factor in, as was said earlier, whether or not you're a night owl or a morning person. But this is one way that a mutation can affect splicing that can affect our health, or at least our behavior. Alternative splicing is important. Okay. Last point about eukaryotic gene expression regulation, lactose intolerance. So normally, as I've said last class or two classes ago, we start out as infants, all of us, being lactose tolerant. We're meant to ingest, well, evolutionarily speaking, we're designed to ingest only milk during the first part of our growth. And eventually by adulthood, it's typical, it's wild type, I don't want to say it's normal necessarily, it's usual that we become lactose intolerant. And that's because this key enzyme, lactase, that breaks down lactose, milk sugar, gets turned off when we're adults. The gene expression goes down, either transcription or translation. Our bodies just don't produce that enzyme that breaks down the sugars in milk anymore. That's typical, that's common. Some would say that's wild type for humans. How does this happen, do you think? It's an abstract question. There's not necessarily a correct answer. But how can one organism with the same DNA, right? This is one individual over their lifetime. Same DNA sequence in all their cells, early, young, later, when they're old. How do we start life with a gene turned on, lactase, and end up later with it turned off?
Mutations?
Same DNA sequence, not mutation.
Maybe a sequence that happens during puberty, there's a chance that there's a Y brain cells, count lactate.
Well, there has to be something physical to turn off. We just studied transcription and translation regulation, gene expression regulation. Something physically has to happen to the DNA around this gene to turn off gene expression or to turn on gene expression. So it's repression, it's activation, but how does that change over life? Wouldn't you assume, I would assume, that if you start off life making a protein, how are you going to get a chronological change, right? This is time passing. This is not cells changing in DNA sequence. You start with the DNA sequence that you were born with, your cells all have the same DNA sequence later in life. So what's happening between start and end is not mutation. Exposure?
How often do you get exposed to lactose?
Use it or lose it. Okay, so that's in your gut though. How would you imagine, I'm not saying that's wrong, I'm just asking for more detail. How would that translate into gene gets turned on or turned off? Exposure.
Regulated by growth hormones or something?
Regulation by some sort of hormones, so you ingest a sugar.
Stop making it, turns on lactate.
Okay, so we'll go with use it or lose it. Other comments though, I want to make sure everybody's heard here. Other comments about how this might happen? So a fair point to make. We initially, as infants, produce our own lactase to break down milk sugars. But then when you're an adult, you can ingest probiotics, bacteria that live in your gut, that will do that job for us. So there are two different ways to be lactose tolerant. One is to have the organisms in us to do it for us, but we start out life producing lactase ourselves. And that's more what I'm trying to get at is the gene expression regulation. But you're absolutely right. There is more than one way to skin this cat.
Even something like introns and exons, you might get a splice if it's over time. The splicing might favor one side after X amount of years.
So what we do know about what happens to us over time, and you've seen this video, I think. I assigned it, I'm pretty sure I assigned it, but not many people watched it. Epigenetics.
It's not like that's why people with lactose intolerance feel like
But it still requires mutation.
So there's no mutation?
No mutation. No, it turns out this is epigenetic change. So this is how we start life with one phenotype and can end up, even though it's the same organism with the same DNA in all of their cells, you wind up with a different phenotype or different trait. Epigenetic change. So chemical modifications to our DNA that are not mutations. It's not changing A to T or A to C or G to T or anything like that. How do epigenetic changes affect gene expression? For those of you that did watch that video a couple of weeks ago, does anybody remember? It's methylation and acetylation of histones. So recall that DNA is wrapped around those histone molecules, proteins. So we've got DNA, we've got histones forming nucleosomes, and over time, exposure to certain things like pollutants, that's not necessarily the case for lactose tolerance, but we're going to talk about a great case of exposure to pollutants and how that affects us in the next class. Over time, methylation or acetylation of these proteins leads to compaction of the nucleosomes. And this makes us go from having what sort of chromatin to what sort of chromatin. We start out, open chromatin structure is euchromatin. And then what happens after epigenetic change? that becomes compact or heterochromatin. And I want to make it really clear, though, that this is not always a one-way street. So epigenetics does not always mean turning euchromatin into heterochromatin. It can go either way. So epigenetic changes, chemical modifications to the DNA, to the histones that wrap the DNA, can cause euchromatin to become heterochromatin or heterochromatin to become euchromatin. So those chemical modifications can turn genes off. That's heterochromatin. Or those chemical changes to histones can turn genes on. So if you haven't watched that epigenetics video, or if it's been a while, I suggest go back and re-watch it. Now, occasionally, we get big baby syndrome, where you have an individual who happens to maintain expression of lactase into adulthood. So they're baby-like in that their body still produces lactase, but they're adult. This could be mutation. It could be epigenetic change, but the story I'm going to tell you about today is mutation, and how some adults maintain lactase persistence or the ability to digest milk products on their own into adulthood. Now, how long have humans been around? Modern humans, Homo sapiens. Tens of thousands of years. These mutations, geneticists have identified, have only been around. There's mutations that allow adults to produce lactase, only been around a couple thousand years. So a tiny fraction of the amount of time that Homo sapiens has been around have adults, some adults, not all adults, been able to produce lactase. And in particular, we're going to focus on a mutation C to T, here in the middle of this drawing, at minus 13,910. Remember that, minus 13,910. But what does it mean? What is a minus 13910 mutation? What does that tell us? How many? It's a location. It's how many nucleotides from what? Yeah, it's nucleotides from the transcription start site, the +1 nucleotide that we learned about a couple weeks ago when we talked about transcription. And it turns out this particular mutation, which originally was C, but some of us are, in this case, is a heterozygote, for example. We've got a CT heterozygote. One of their chromosomes has a C, one of the chromosomes has a T. This nucleotide is 13,910 base pairs away from LCT, which stands for lactose tolerance, or lactase. That's the gene that encodes that protein that we need to be lactose tolerant in adulthood. So this mutation is over 10,000 base pairs that direction of the gene. It's actually in an intron of the MCM6 gene. So this mutation is inside another gene. That way. Is the T mutation, talk to a partner for half a minute, and then we'll come back together. This is a heterozygote. This individual is lactose tolerant. Which allele is dominant, C or T? So talk to somebody else, don't say it out loud yet. Turn to a neighbor, convince them you're right. Is C or T dominant? Which of those two? Okay. So what do you think? Is it dominant or is it recessive? T. Okay, so why would T be dominant? Can you explain what?
Unless A is dominant.
Unless, so you've got a heterozygote and T is the mutation, C is the wild type allele, T is the new one. You've got a heterozygote, they're tolerant. If they're lactose tolerant, what does that mean they're doing at their lactase gene? They're producing lactase. So that means lactase is being produced, this enzyme that breaks down sugar. So T is this classic crosses. If you want to know an allele is dominant, you make a heterozygote and you find out what their phenotype is. The phenotype is they produce lactase. They're lactose tolerant. Therefore, T is dominant. Because only one copy of T is necessary to produce the tolerance trait. If we only saw lactose tolerance in individuals that were T over T, then it would be recessive. So it's a dominant mutation. Basically, it just means that only half of your copies of the lactase gene need to be turned on to produce this enzyme to give you some level of lactose tolerance. Kind of makes sense. If you've got milk sugars in your gut and you're making half as much lactase as a homozygote, at least you're breaking down some of the sugar. So it's a dominant mutation. And this is just a chart of that's reiterating that this mutation is in an intron in a different gene. So this is all the MCM6 gene. It's about 10,000 base pairs away from the gene that this mutation actually regulates. 13,000 base pairs away. This is not a promoter. It's in an intron. This is not the promoter of the lactase gene. That's down here by lactase. So what sort of mutations, how could mutations that far away from a gene affect transcription of lactase? What might that actual one base pair mutation do? It's an intron way over there, different gene.
Keep the sugar the same.
So this is a mutation that's not in the gene, so it's not going to affect splicing, because if this was going to affect splicing of lactase, it would have to be in the lactase gene, in its exons or introns. If it was going to affect the lactase enzyme, it would have to be in an exon of the lactase gene, LCT. So what's left? How can a mutation somewhere else in the genome affect whether or not lactase gets turned on? It affects a transcription factor binding site, an enhancer. We've talked about enhancers before, that they can be tens, hundreds of thousands of base pairs away from the gene they regulate. And it turns out that this one mutation, I'm going to circle it here because they didn't mention it, but that's the minus 13910 single nucleotide polymorphism, C to T change, improves a binding site for a transcription factor. It's a transcription factor called OCT1, but it has a specific DNA sequence that is its normal binding site. And when you make that mutation from C to T, that creates a better OCT1 binding site here in the intron of the MCM6 gene that's 10,000 base pairs away from LCT, lactase. So there's an enhancer, a DNA sequence that regulates transcription of lactase that's in a different gene, 13,000 base pairs that direction from lactase. So mutations that affect gene expression literally can be basically anywhere in the genome. For any particular gene, you can't just look in the flanking sequence, the immediate sequence, especially in the promoter, the 5' direction, that's not where all of the gene expression regulation for a gene happens. Enhancers can be anywhere. And I talked about this some in the pre-lecture video, not for today, but from last week, talking about eukaryotic gene expression regulation in the GAL system, galactose. We talked about GAL4, GAL80, and so forth. This is basically the same idea, except I'm just going to scratch out some things. In this case, we're not talking about GAL4. What's the enhancer, the transcription factor that's bound to the enhancer? It's OCT1. Its UAS, its upstream activating sequence, is in an intron of, I don't remember what the name of that gene was, MCM something, MCM6, I think, which is 10,000 base pairs away from not the GAL genes, but what gene are we talking about in this case? Regulation of lactase, LCT. So same picture, just different molecular players. But the idea is the same, is that despite this huge distance between the promoter of lactase and the gene sequences and the proteins that regulate it, that's able to happen because DNA is flexible. And you can bring parts of DNA enhancers really close to the promoters of genes by the DNA simply bending, looping, which it does normally. That's why this is sort of distance irrelevant. Enhancers, sequences that transcription factors bind to can be millions of base pairs away. They could be on a different chromosome. Thoughts, concerns?
I mean, if something like that is the case where it's, well, I understand when it's far apart, the DNA can bend as it's being transcribed. But if it's on a different chromosome, the chances of it affecting it, what does it do to Brownian movement, or?
So it's a great question. Could it really be on a different chromosome, basically, is the question. So what are some of the obstacles to having a regulatory element for a gene be located on a totally different chromosome? You mentioned the key one, but I want to know if there are others people can think of. What's the difficulty of having a regulatory element for a gene be on a different chromosome?
Probability of them crossing paths?
Yeah, it's basically, like you said, so what's the likelihood? The nice thing about having a gene next to its regulatory element, like an enhancer being close to the promoter, is that that interaction is likely to happen because the enhancer is near the gene. So you're likely to get physical interaction between the enhancer and the gene it regulates. What happens in a different scenario, in the nucleus, if you have an enhancer over here on one chromosome and the gene it regulates is over here, right? This is basically just a physical issue. The farther apart two things are, the less likely they are by chance to come in contact with each other. So you're absolutely right. That's a good point. The really cool thing about the nuclear genome, which we might not have talked about though, is that chromosomes tend to stay in the same parts of the nucleus, which is bizarre. And I definitely encourage you to look at this information after class. But when chromosomes arrange themselves in the interphase nucleus, they do actually tend to stay in the same parts of the nucleus relative to other chromosomes. It's not just random. It's some of it's random, but some of it's not. And there are actually domains, parts of chromosomes that tend to stay closer to the periphery of the nucleus, and other parts of chromosomes that stay closer to the center of the nucleus. And this is repeatable, cell to cell in the same individual, the same species. Who knew? So chromosomes actually unpack themselves during interphase in an ordered fashion. It's not just all parts of chromosomes go wherever they want when they become euchromatin during interphase. So this is from, all of this information I've been telling you about is from this study that's listed there at the bottom. It's a 2011 manuscript that found this mutation in lactase, or in MCM6 rather, that controls lactase production. And part of this manuscript involves this map. We're looking at the distribution on the bottom here. There's a scale here that tells you frequency. How frequently this mutation is found. So which parts of the world you're more likely to have that SNP that keeps you lactose tolerant. So just think about this as a map of lactose tolerance. If you're from a region of the world where there's red, you're more likely to have this mutation, you're more likely to be lactose tolerant yourself. Or, if I could spell, you would see that you're more likely to be lactose intolerant if you're from one of the regions where there's a lot of SNPs, not as frequent. Most of the people in the population have the wild type version. And look, you can kind of see how lactose intolerance is wild type. Most of the globe is covered with blue. Right? Most people in the world don't have that T mutation. Where is the T mutation most common? Western Europe, according to the map. If you're most likely to be lactose tolerant if you're from, looks like the British Isles, basically. Okay. Now contrast that with what's on the top. What's that a map of? Lactase persistence. That is, individuals that are, regardless of whether or not they have this specific mutation, just individuals that are known to have the phenotype. They're able to digest milk sugars. Pretty good overlap other than Western Africa, but you see that Scandinavia and the British Isles, some parts of the Middle East, some parts of India or near India, there are some little pockets of individuals around the world that are lactose tolerant into adulthood. So this mutation only explains a small fraction of the people in the world being lactose tolerant as adults. So there must be some other changes in other populations that allow them to be tolerant.
Could part of the relatively higher persistent, well, slightly higher persistence in the presence of the mutation just be like the lactase staying in the diet, but not more being produced and then sick, it wipes it out and you don't produce anymore?
Yeah, right. So this maybe gets a little bit back to the earlier comment of why are you lactose tolerant? Is it because you've got gut microbiota that break down the lactose for you, or is it genetics? And it's not clear that that's necessarily the case for the top chart. So that might have to do more with what you eat and your environment than genetics, but it might not too. So this was a, go ahead.
So if you are going to argue like exposure.
Ooh, let's argue, yeah.
Not so long ago, I saw a couple documentaries that right along where like the equatorial places where it's all red, a lot of people that are nomadic, so they rely heavily on herds, mostly goats or what have you, or cattle, and one of their main sources is,
Or camels.
or one of their sources is milk from their herd.
Right. So that's why, and that's a fantastic segue. Thanks for mentioning that. That's why I'm particularly interested in this research paper from 2011. Normally genetics papers are pretty dry because they just talk, well, I'm sure you understand. You can imagine if you haven't read a genetics paper before. This one's kind of interesting though because it got into a little bit of sociology and anthropology. So they noticed that these mutations, this one mutation, the -13,910 mutation, has been around about as long ago as human populations started domesticating cattle, goats, camels, and other organisms for producing milk for us to consume. So they ended this paper not with some sort of dry genetic hypothesis or conclusion, but rather they came up with two different hypotheses, and I'm going to ask you about this in a second. We're probably not going to get much into genetically modified organisms today, but they came up with two different theories for how lactose tolerance might have occurred and why there's this overlap between where domestication of animals for milk production occurred and where this mutation occurs. The first hypothesis up here, I tried to make a graphical representation. I don't know if it's going to work for you very well, but let's try it out. The first one, the culture historical model. basically says that humans started dairying, so this is early in time. There were cows, camels, sheep, goats, whatever, domesticated for production of milk, and nobody at the time was able to consume and be healthy or be happy about using milk as adults in the population at the start. But the presence of that food source over time, once you had that mutation here, LP, lactose persistence, as soon as one individual in the population got that random mutation that allowed them as an adult to use milk, then what happens to that mutation? It's inherited more preferably by other people because it gives you an advantage. So over time, so this hypothesis, culture historical, suggests that culture happened first. The dairying practices happened first, and that provided the opportunity for when somebody got the mutation that let them use the milk in their digestive system, all of a sudden now it spreads, and everybody in the population over some number of generations inherits that beneficial trait. The second hypothesis is basically the opposite. That there were, in some human populations, this mutation already existed. Right? So not everybody in this early population has lactose tolerance, but some individuals have this mutation already. That makes that human population perfect for starting dairying. So you have the mutation first, and it's in the populations of humans where people are already naturally lactose tolerant as adults that you see the cultural practices emerge. That, okay, in those populations, that explains why they started milking cattle and sheep and goats and whatever, because they already had those mutations that let them digest that resource as adults. So we don't know, of course, we can't go back in our time machine. We don't know which of these is true, or which, if either. Could be neither of these is what happened. So we've got the culture historical model, and we've got the reverse cause model. So what I'd like you to do, please, we'll take most of the rest of class, if you like, to do this. We'll close in about 5 or 6 minutes. But to go to Socrative and think about that sort of question. So I'm going to let you log in. I'm going to flip back to that slide so you can see those two models again. I'm interested in knowing which you think is more likely.